Hi, everyone. I'll explain what you're looking at in a minute, but this is the weekly announcement for week one, but going into week two. So, what I will do with the weekly announcements, I'll start out with I will cover some concept that I feel like by looking when I look what I'm looking at right now are the statistics on each item in the assignment called Chapter 20 Interactive Assignment. Okay. So I go through and look at the analysis of each question in the assignments, and I pick something that I pick a topic that I think that you guys maybe need a little bit more instruction or clarification on. And then sometimes, some weeks, the topic that I'll cover is something that you guys shared maybe through an email or through the weekly Q&A discussion forum. But anyway, I always start out these weekly announcements by going over some concept from the previous week. And then I will also make sure you know everything you need to do for this coming week for the assignments that are due on June the 3rd, Monday, June the 3rd, by the end of the day, which is 11:59 p.m. But I do want to say congratulations to making it through the first week and great job. There was very high participation from the class, and almost all assignments were completed. And even those who didn't complete their assignments, I got an email, you communicated with me. So great job. Just remember that most weeks you're going to have two chapters. So last week was kind of getting you started and getting you used to Connect and used to Moodle, the Moodle course, that kind of thing. But now you're going to have two chapters each week, so double the assignments that you had last week, okay? So I wanted to talk to you about the Gram-Stain procedure on bacteria. And first, just to review, the Gram-Stain procedure, what it actually does, and okay, this is an animation. I don't want to show an animation. I just want, hold on. I just want a list of the reagents that are used, which is right here. Okay, so the Gram-Stain procedure, the purpose of it is to stain bacteria so we can see them under the microscope, because otherwise, if you don't stain them, that they're invisible under the microscope. They're very hard to see. So there are many different types of stains, and if you ever take microbiology, which is Bio 275, that class, you learn a lot of different types of stains. There's one I know called the acid fast stain. But anyway, there are different stains that you can do, but the purpose of staining in general is so that we can see the bacteria under the microscope. So the Gram-Stain focuses on two types of bacteria based on the composition of their cell wall. So if the bacteria is called gram-positive, their cell wall has a very thick layer of peptidoglycan, and you can see that in this picture here. This is a picture of a gram-positive bacterial cell wall. And you can see the cell wall is very thick. It's thicker even than the plasma membrane. Here's the plasma membrane underneath it. And it's also showing you the first two reagents or stains that are used in the Gram-Stain procedure. So when we do the Gram-Stain procedure, we start out by staining the gram-positive bacteria with the thick peptidoglycan layer in their cell wall. And then we stain the gram-negative bacteria, and they have a thin peptidoglycan layer. But the first two reagents that are used, the first one is called crystal violet, and it's a purple color. And what happens is that cell wall of the gram-positive bacteria will retain the crystal violet stain. In fact, when you add crystal violet, all of the bacterial cells, whether they're gram-positive or gram-negative, will absorb that stain. But what happens is, unless you add iodine, the iodine helps to lock in that crystal violet stain, so it won't wash away, okay? if you don't add the iodine, the crystal violet stain is going to wash away. It's not going to be absorbed into that peptidoglycan layer. And so it's just going to wash away when you finish the gram stain procedure. So crystal violet is to stain the gram-positive cells. Iodine is to lock in the crystal violet stain. And then the last thing that you do, well, then you have a step called the decolorizing step. You add ethanol, ethyl alcohol. And what that does is that takes the crystal violet off of everything except the gram-positive bacteria. It removes it from the slide, so you're not looking at purple bacteria on a purple background. And it removes the purple stain from the gram-negative bacteria. So these are the reagents that are used. Crystal violet stains the gram-positive bacteria purple, and the iodine is necessary for that crystal violet to lock into the peptidoglycan layer of the gram-positive bacteria. The ethanol is the decolorizer, and then safranine is the red stain that causes the gram-negative bacteria to appear pink. So here's the question that a lot of people missed even after a second time, and this is the one I really wanted to go over with you. It says, it's from this same video, and it says, after performing a gram stain of a mixed culture of gram-positive and gram-negative cells, you shout, "Oops, I forgot the iodine step." What would you expect to see if you observe the slide under the microscope? So in other words, what you have done is you've added the crystal violet, you've added the ethanol, the decolorizer, and you've added the safranine, but you forgot to add the iodine and lock in that crystal violet to the cell wall of the gram-positive bacteria. Okay, so basically, since you did not add the iodine, when you do the decolorizing step, all of that crystal violet's going to wash off of the bacteria and the slide, and it's just going to be gone. So what's the last thing you do? After the decolorizer, you add safranine. Well, safranine is going to make the cells appear pink. Safranine actually is absorbed into both types, the gram-positive and the gram-negative. So what you would see is pink gram-positive cells and pink gram-negative cells, because both types of bacteria would absorb the safranine stain. You just can't see it. Usually you can't see it in the gram-positive because that crystal violet is so much darker of a color than the safranine. Okay, so that's the reason the answer is pink gram-positive and pink gram-negative cells. Okay, so I hope that helped you. Now, the next thing that I'm going to do is go to our class on Moodle, and let's talk about this coming week. That's the assignments that are due on June the 3rd. All right, so over here on week 2, once it comes up, the 2 chapters we're going to cover are chapter 21 on the protista and chapter 22 on the fungi. Protista and fungi both are in the domain Eukarya. The domain Eukarya. And the domain Eukarya is composed of organisms whose cells have a nucleus and membrane-bound organelles. So there's no bacteria, and there's no archaea. It's only eukaryotic cells. So chapter 21 is on the kingdom Protista, which includes algae and protozoans. And protozoans are microscopic, but they're much larger than bacteria. And it also includes chapter 22 on the fungi, kingdom fungi. And the other kingdoms within the domain Eukarya are kingdom Animalia and kingdom Plantae. So if you look in you've got your learning outcomes listed, and you can read through those. For the instruction, of course, it's optional. I always recommend you do one of these, and then if you're having a hard time, maybe you need to do both of them. But just one is usually sufficient. take notes from some source, though. Either if you like to read the textbook, take notes from the highlighted portions in yellow. If you would rather watch the recorded lectures and take notes, same thing, take notes on the highlighted portions of the slides. And those are not graded assignments, but they're that's your instruction portion of the class, and I highly recommend that you do them. Even if you don't, though, if you do the Chapter 21 Smart Book, that is graded, and I did notice a few of you did not do it, and it's probably because it takes a long time. But that is part of your instruction, is to complete that Chapter 21 Smart Book activity. And you also will find, if you do the Chapter 21 Smart Book, it helps you learn the material. Some of you will be able to get away with only doing the Chapter 21 Smart Book. From this section on instruction, only completing that Chapter 21 Smart Book, which is a graded assignment, and it's going to be listed in the Moodle gradebook as a homework grade. Then you go on and do your activities in Connect. So far, your first assignment that's graded has been the Chapter 21 Smart Book in Connect. Then you move on and you do the virtual labs, and this week you're going to have virtual labs called Diversity of Microorganisms, Diversity of Protista, and the Euglena Wet Mount Lab. That shows you how to make a slide of living protozoans called Euglena. And then you're going to complete the Chapter 21 homework and the Chapter 21 interactive assignment. Then you're going to move on and do the Chapter 21 homework quiz, which is just 20 random questions pulled from the homework assignment. But it's graded as a quiz grade. So the labs are graded as a lab report grade, and then the homework, interactive assignment, and smart book are in the homework section of the grade book. And the homework quiz is graded as a quiz grade. Then you're going to move on to Chapter 22 and do the very same things. And just make sure you do the graded assignments are the Chapter 22 Smart Book, Diversity of Fungi Virtual Lab, Chapter 22 Homework, Chapter 22 Interactive Assignment, and Chapter 22 Homework Quiz. If we scroll down so that you can see what else is in on the Moodle page, so all of your assignments for this coming week are due June the 3rd, which is Monday. So what you see underneath that section that lists all your assignments and everything, overview section is what I call it, is you see the McGraw-Hill campus link. So you can click on that and go directly to McGraw-Hill. And that's the best way to do it, is just access it from here. Go to Moodle first, click on the McGraw-Hill campus link. And then, of course, after you do that, if you want to bookmark it and just go to it from your laptop, that's fine. But then we have the slides posted for Chapters 21 and 22, and then the recorded lectures. So, and I think that's all. Oh, no. Okay. I highly encourage you to participate in the weekly Q&A forums. Now, underneath this, I'm not sure if I made the rest of this available to you or not. I think maybe it is. This is just additional videos that I got from YouTube on the material that's covered this week. Totally optional. But the ones above here, the ones that look like this and that have an L in the corner, that means they're recordings that I made. So I just went through the slides and recorded the lecture, along with the slides, and explained the slides to you. So I try to keep these little recorded lectures to like 15 minutes each, so that you can stop and take a break before you go on to the next one. But, that's so that you can have, some instruction, I guess, So anyway, that is it for this week. If you have any questions, remember to use the weekly Q&A for questions about the chapter content. And especially if it's a question that could impact your other classmates. And then if you have any questions specifically about your grades or you've got a technical problem that's going on with Connect or Moodle, any of that kind of stuff, you can email me directly. All right, so I hope you guys have a great week and a great weekend. And just let me know if I can do anything for you, if I can help you in any way.